Hello, I'm Tony Olszansky. I'm a medical oncologist at Fox Chase Cancer Center. I focus on the treatment of lung cancer as well as skin cancers like melanoma and squamous cell carcinoma. I also lead the research program here called the Phase 1 Oncology Research Program. Phase 1 trials offer patients therapies that are not yet approved by the FDA in an effort to try to help them with their cancer. Today, I have a presentation on the importance of pharmacogenomics. Pharmacogenomics is really the study of somebody's individual genes and how they might influence the way that medicines work in the body and specifically on specific cancers. So that's what we're going to talk about a little bit today. So I'm going to just move through some of the slides here. And I'm actually going to get to a couple important points of the presentation, just moving through some of the questions that I have on the screen that I think that a lot of people find pretty amazing. For example, how many genes are in the human genome, and we think there's about 20 to 25,000 genes. And when we think about how many parts of DNA are in a single somatic cell of the body, any cell in your body, there's about 3 billion base pairs of DNA. And if you look at all human genetic variability, that is, if I compared a person who is in the United States with a person who is in Africa, with a person who is in Europe, for example, we're all extremely similar. In fact, our genetic background is 99.9% identical, which is pretty amazing. And then also amazing, I think, to me, is that the human body is estimated to hold about 45 trillion cells. And I show this slide because I think it's important to recognize that genes play a huge role in not only the people that we are, but also the cancers that might develop and how they can be treated. Cancers develop because of mutations in any one of these many, many genes that we house. So we're going to think about some goals of pharmacogenomic testing. We primarily use pharmacogenomic testing to avoid adverse drug reactions. We want to maximize how drugs work in a population. So we maximize drug efficacy. We select responsiveness. We want to find patients who are more likely to respond to a given patient. And we assess hereditary risks. So I'm going to go through just a few other things here and then get to the main part of what I wanted to really spend my time on in this presentation. Historically, we've known about genes for a long time. In fact, Theodore Boervie, who lived in the 1800s and died in 1915, he said that tumorigenesis, or the development of tumors, results from a particular incorrectly combined chromosome array. This is the cause of the tendency to proliferate, which is passed on to all descendants of the original cell. So all the way back in the late 1800s, early 1900s, we recognized that cancer was because of some kind of genetic defect in a cell that then allowed that cell to live on, even though the body was trying to kill it. That's not to say that all cancers come from mom and dad, but rather that we can develop cells in our own body that have genetic changes. So when we think about, again, pharmacogenomics, we really think about how drugs, that is the pharmacology, affects to genomics, or the gene. The drugs, the gene, and the environment all interact together to produce either responses in patients, adverse drug reactions, or we can select patients based on what we know and come to different outcomes. But personalized medicine, which is what we often talk about in oncology, means a lot of different things to different people. Some people have quoted that they think that they have their own medicine. Some people have suggested that they just see one doctor all the time. But that's not really what doctors think of when we think of personalized medicine. What we think of personalized medicine is that we have specific drugs or targeted clinical therapy. And through research and through screening of the genome, we can personalize therapy so that it is given to a patient that has a specific target in their tumor. So there's a lot of promises of the pharmacogenomic therapy. We recognize already that everybody's therapy is not true for everybody's body. Therapy changes depending on us, changes on our body. Again, we talk about the pharmacogenomic goals, and we want to try to avoid adverse drug reactions. We want to maximize the drug efficacy. We want to select the patients who are going to respond. One of the giants in medicine, Sir William Osler, back in 1892, quoted that if it were not for the great variability among individuals, medicine might as well be a science and not an art. We really have to take all of the information that we have for a given patient before we can select his or her best therapy. And that's what really Dr. Osler was getting at all the way back in 1892. And what's nice about living here in the 21st century is that we have so much more information now for patients. We understand a lot better how we might be able to best treat them. We have a lot of drug metabolism efforts when we think about genetics and specifically changes in genetics or genetic polymorphisms. And two drugs that we commonly use in oncology, one of them is called 5-fluorouracil, which you can see here in the middle of the slide, and another one called irinotecan, are heavily dependent upon how well a body can metabolize those drugs. Some patients have a specific deficiency in a gene called DPD, and that can lead to enhanced toxicity if we give them 5-fluorouracil. So again, knowing about the makeup of the genes of a person can help us determine what therapy might be best for them. Likewise, if a person has a UGT1A1 mutation, it can cause irinotecan, another common chemotherapy, to have enhanced toxicities. And so we'll sometimes check these genes prior to applying these chemotherapies for patients. You might be recognizing a different medication here called codeine. Codeine is actually a medication which is available across the world. It's a pain control medication. As it turns out, codeine is metabolized by an enzyme called CYP2D6. And about 10% of the population, as we understand it, don't have that particular enzyme working very well in their body. And if it doesn't work, codeine won't work. And so it can actually lead to decreased effect. So I hope with this slide that I've also convinced you that the genetics of a person, the way the genes are laid out in that given person, can affect how drugs work. Either they work less effectively, maybe more effectively, maybe causing more toxicity as well. We know that genes are important. We have some genetic tests that we apply specifically to cancer, like the Oncotype DX test. The Oncotype DX test is looking at 21 specific genes, and it looks at their expression, and it helps predict for an oncologist whether or not chemotherapy will be beneficial in patients with certain types of breast cancer that we call ER-positive, HER2-negative. It's not specifically looking for mutations or anything, but it can certainly inform an oncologist about what medications might be best to give a person. We have similar studies in colorectal cancer. In this study, we were looking at what a gene called BRAF might have regarding the influence of patients with colorectal cancer. And we recognized that if a patient had a mutated BRAF, a gene which has been changed in the cancer, that patients generally did not do as well as those patients who had no such change in that gene. And that again helps us provide new therapies for patients or provide therapies on a clinical trial in an effort to try to get over this hurdle in some patients who have this BRAF mutation. We do a lot of testing for next generation sequencing these days, trying to figure out what all the genes are. We can do this on a very, very small amount of DNA. We only require about 10 nanograms of DNA. We can get that through a small biopsy. Therefore, we don't need a lot of tissue. And we can do this sometimes just with some liquid samples called fine needle aspirations or FNA samples. This multiplex testing or next generation sequencing tests tens to hundreds of genes today. And so we can get a lot of information from a small test. And it actually gives us information fairly quickly. After we get the biopsy, we can usually have results of the test within 2 to 3 weeks. And this can really have a lot of implications in treating certain diseases. This slide looks at patients with lung cancer. And if you look at the slide, you can see that in the light gray part of the graph here, patients do not have any known mutation that we can test. But if you look at the red and orange part of the pie graph here, you'll see a gene called the EGFR gene. The EGFR gene is mutated in about 17 to 20% of patients that have a form of lung cancer called adenocarcinoma. And what's exciting about that particular medication is that that particular mutation is that we have specific medications that you can take orally that target that gene mutation and can drastically change how well patients do with lung cancer. Medications like erlotinib and osimertinib have revolutionized now the treatment of patients with EGFR positive lung cancer and have allowed them to live normal lives with relatively limited toxicity from the drug. And this is really important. So as you can see, cancer genetics can really help us in figuring out what drugs we need to apply for given diseases. This particular slide shows certain disease examples where this information can be really quite helpful. I wanted to review very quickly with you some clinical pearls that we've understood as medical oncologists and why we think mutations are so important. This cartoon is a pretty simplified cartoon of how we envision a cancer cell, the receptors on the surface of the cancer cell, and the downstream signaling that occurs all the way down to the nucleus, which here you can see represented in a light blue color. On the cell surface, you have receptors. We call them receptor tyrosine kinases. They're in the blue, the red, the green. And below that, inside the cell, in the interior portion of the cell of the cytoplasm, we have a number of different molecules being represented there, or proteins. And those proteins interact with each other. And we can possibly turn off one pathway. So for example, I could give somebody a BRAF inhibitor, and that would knock off the RAF pathway so that it couldn't have any downstream effects. But that could lead to redundancy or upregulation of the pathway that you see on the left-hand side, which is the PTEN-PI3 kinase AKT pathway. And so we have to understand these fairly complex pathways when we're applying medications for patients. We recognize that one pearl from this particular slide suggests that tumor suppressor gene inactivation can lead to activation downstream. So we really have to be pretty smart as oncologists to understand the entire complex human system before we apply these medications. This slide goes on to show how this is best applied in a given patient. On the upper left-hand corner, you can see a couple PET scans of a patient taken at different time points. And what you recognize in this PET scan is the patient had certain areas in their body that were cancerous, and they were given a medication, and that medication actually helped them for some time, and then the cancer began growing again. We got to sequence them again, take another look at their tumor type, figure out what other medications might be helpful for them, and then applied that again and got another good response. So that's what that PET scan is trying to demonstrate, that we can see responses, and then we can see resistance build up to the drugs that we apply. But with the knowledge that we now have, we can get more information from the tumor, why that resistance is actually happening, and then apply new medications. We can also recognize that some of our medications are so effective that they can literally obliterate cancers really quite effectively. Unfortunately, what we're also recognizing now is that sometimes when we stop those medications abruptly, if a patient is having side effects or if they have had new disease, progressive disease, their disease can start growing very, very quickly, suggesting that the disease is somewhat addicted to a given pathway. And so we take this information and use it to try to figure out which is, in fact, our best next therapy and when do we actually apply that. So the timing then of getting medication is really important when it comes to oncology. I'm not going to review this slide too much. It just talks about a little bit of what we've already said, which is if we continue to inhibit a protein, sometimes that can be beneficial for a long period of time. As you may understand about cancer, when we develop cancer as humans, we often develop in an organ of our body So for example, I might develop lung cancer that would start in my lung. But that lung cancer doesn't have to stay in my lung. It can move to different parts of my body. It could move, for example, to my liver. So it becomes important for us as oncologists to understand if the cancer originated in the lung and we understand its genetic composition, and then it moves into the liver, does the genetic composition change in any meaningful way? Because that might influence, again, our therapy. In this particular study, we were able to prove that there's a high concordance of cancer in where it starts and where it actually moves. So that is to say that if I developed lung cancer and I understood the genetic signature, if it did move to the liver and I did another genetic test, there's a very high chance that it would still be concordant, that it would look the same. And that really helps us because we can continue to apply the same medication regardless of where the tumor is in the body. There's obviously some caveats about the way that I'm presenting this and the way that I say that, and it's actually a lot more complicated than that. But this gives you some information to suggest that as cancers move around in the body, they have some similarities to where they came from. And that can be very helpful when we're thinking about applying certain tests. We've learned a lot through this next generation sequencing, and in this slide, I show you some sequencing that we do on a protein on the upper part of the panel called KRAS. We often look for mutations in something called exon 2, which we can see in about 40% of patients. What we've recognized through this big testing that we do, the next generation sequence testing, is that we now have other mutations that we need to be concerned about. And so there's at least about 17% of additional mutations in both KRAS and NRAS, which can influence which treatment choices we might have for a given individual. So signal transduction pathways are complex and redundant and very important for a medical oncologist to understand before we apply any medication. So I'm just running through a couple slides here, trying to get through to the conclusions here of some of the stuff that we've talked about. We've already talked about the fact that cancers that move from one part of the body to another part can remain very similar. This cartoon actually shows that tumors are somewhat genetically unstable, and over a long period of time, as that tumor is moving through the body, it can start gaining different attributes, and this can lead to therapeutic resistance, and in fact, resistance to certain therapies might pre-exist before we ever apply them. New clones of cancer can have different mutations, and so we have a number of different learning points that we can draw from this. And that is that genomic instability or instability in those genes can lead to different subclones. And we have to be, again, aware of those subclones and how we apply them. This graph basically shows how we applied that knowledge in a patient with EGFR-resistant colorectal cancer. In this particular study, 24 patients who were treated with that particular drug all progressed within 6 months, and we started recognizing when resistance was occurring. And really, through the use of genetic testing, we could see that they started becoming resistant even before a CAT scan or a radiographic scan showed that they had new disease. So that's important as well. We also finally recognized that certain genetic mutations are very different depending on what cancer they arise in. So I can have a patient with melanoma, for example, who has a BRAF mutation, and I can apply a medication, and they can have a chance of a 70% response rate. But if I do the same thing in a patient with the same exact mutation in colon cancer, their chances of having a response is probably only about 5%. So we recognize that it's not only about the genes, but it's also about the cancer. And so some of these genetic targeted therapies are very tumor dependent. So I'm going to move to the conclusions of this slide deck. We really do continue to discover new ways of testing patients. We now have these roadmaps or these strategy roadmaps that can sort of list for us all the different mutations and list for us therapeutic choices or give us more information so that we can pick the therapeutic choices that might be in the best interest of a given patient. This can also be very helpful if we're picking therapies for patients for clinical trials. Some of the medications that we use today are not yet FDA approved, They're still going through testing, and through that avenue, we often use clinical trials. And these types of tests, these genomic tests, can identify certain subsets of patients that can benefit heavily from clinical trials. So, in summary, early on, metastatic lesions appear to be very similar to primary lesions, as we've already pointed out, but genomic instability can help explain the emergence of resistance. Just because a patient has progression on a targeted therapy doesn't mean we should necessarily change that therapy. Sometimes if we continue that therapy, we can still see good results. We can see signal transduction pathways activate when suppressor genes are non-functional, as you can see here, and patient selection is often critical. And I guess that's how I'll really conclude this talk, is that when it comes to your care as a patient with cancer, the patient selection makes all the difference about what therapies we're going to give, about how much we understand in your body, both from all the medical information that we gathered, the radiographic information we gathered, and most recently, over the course of the last few years, the genomic data we gathered. And when we bring all that into the foray in front of the medical oncologist, he or she can select the best medication for a given patient. Well, thank you for your attention to this slideshow, and I hope you found it useful.